Hi guys, it's Diane Gibson, your biology professor. I wanted to make a quick presentation on nucleic acids and protein synthesis. This topic can be a little confusing, so I wanted to walk you through. This is a picture of a cell wall if you zoom in to the molecular level. This is called the fluid mosaic model. You can see that it has a phospholipid bilayer that helps keep the contents inside the cell that are supposed to be inside the cell, and the fluid and everything else that's outside the cell, outside of the cell. It's called the fluid mosaic model because it's a mosaic of proteins. We have lots of little proteins that stick out of the cell wall, and we have little protein channels that help get hormones and nutrients into the cell that need to go to the cell. And also sometimes cells make proteins and nutrients that need to leave the cell and be sent somewhere else in the body. So before I talk about nucleic acids, I just wanted to remind you that there is a difference between a prokaryotic cell and a eukaryotic cell. For this example, I'm going to focus on eukaryotic cells. Eukaryotic cells are cells that have double membrane bound organelles, such as the nucleus or the mitochondria. You might remember the mitochondria from high school biology as the powerhouse of the cell. So in eukaryotic cells, we have double membrane bound organelles. And in prokaryotic cells, we have free-floating circular DNA that is not membrane bound. Because we're talking about protein synthesis, I wanted to focus on eukaryotic cells because I think it makes a little more sense in how it works in a eukaryotic cell. So eukaryotes are multicellular organisms like humans or dogs or cats or whatever, and prokaryotic cells are bacterial cells, so single cellular organisms. So in a eukaryotic cell, so we're thinking of people or dogs or horses or whatever, we have what's called a nucleus. The nucleus is a double membrane bound organelle. Inside the nucleus, we have the DNA. So DNA is formed in the structure of what's called a chromosome. Chromosomes are tightly wound pieces of DNA. DNA is deoxyribonucleic acid. DNA is in the form of what's called a double helix. It has a 5' end and a 3' end. Okay, so when we are talking about transcription, or when we're looking at the genetic instructions of DNA, it is always read from the 5' end to the 3' end. So if this is the 5' end here of DNA, this would be the 3' end, and that's how the structure is formed. So deoxyribonucleic acid has 4 bases. We have adenine, thymine, cytosine, and guanine, where A's bind to T's and C's bind with G's. An easy way to remember that is that C and G kind of look the same. So they kind of look the same, they bind together. Because of the A to T and C to G bonding, we get the twisted double helix form. So this DNA composes the genetic material. DNA is responsible for directing protein synthesis, or the making of proteins in your body. They are the genetic blueprints. They are the instruction manual of the cell, what proteins to make, when to make them, and what those proteins can do. Proteins are very important in the body. They do all sorts of wonderful things, but we have structural proteins, and proteins are also enzymes, so they help speed up chemical reactions. Over here you can see the molecular structure of the deoxyribonucleic acid. So this is what cytosine looks like, and this is what guanine looks like, this is what adenine looks like, and this is what thymine looks like. We are talking about a eukaryotic organism housing DNA in the form of chromosomes inside the double membrane bound nucleus. RNA is ribonucleic acid. It is composed of adenine, uracil, guanine, and cytosine, where A's bind to U's and C's bind to G's. The purpose of RNA is to transport DNA during protein synthesis, or the making of proteins. It is a single chain of amino acids with a sugar phosphate backbone. So, DNA is housed in the nucleus of a eukaryotic organism in this double-stranded form. So it's a really big molecule. It's going to have a really hard time getting out of those tiny little pores in the nucleus. RNA is half the size of DNA. So RNA can come in and out through the nucleus as needed. So the building blocks of DNA and RNA are called nucleotides. That's the nucleic part. So deoxyribonucleic acid or ribonucleic acid. This is a picture of what a nucleotide looks like. We have a sugar, we have a phosphate, and we have an organic base. So the purpose of nucleic acids is to carry the genetic code, or DNA, in the form of RNA to aid in protein synthesis. And the nucleic acid sequence encodes the amino acid sequence that will eventually twist and fold and become a protein. When we are making proteins, your DNA will unwind from the double helix form and sort of flatten out. That way, the RNA can come in and read the DNA sequence. The process of switching DNA into RNA is called transcription, and the process of RNA transforming into a protein is called translation. So DNA to RNA is transcription. We are literally transcribing the DNA. So if you think of 16th century monks having to literally write a copy of a book by hand, they're transcribing the book so that there can be additional copies of that book. Okay, so DNA to RNA is transcription, and then RNA to protein is called translation. So we are translating the RNA into a protein. There are 3 different types of RNA that we're going to talk about. We have messenger RNA, or mRNA, tRNA, or transfer RNA, and rRNA, or ribosomal RNA. So mRNA is the messenger. It carries information from the DNA to the ribosome. So mRNA is a single strand. It enters the nucleus. It reads the genetic blueprint, and it exits the nucleus and goes out into the wilds of the cell. Transfer RNA carries anticodons to pair with the mRNA codons. So we're still having to rewrite the message. This happens 3 base pairs at a time. And then we have ribosomal RNA, which joins amino acids and puts them into a peptide protein amino acid chain. So messenger RNA goes into the nucleus. It reads the DNA instructions. It exits the nucleus. The ribosome will come together in a large and a small subunit and start reading the messenger RNA 3 bases at a time. Transfer RNA will then bring the amino acids over to the messenger RNA and start building that amino acid chain. So there's still complementary pairing of transfer RNA and messenger RNA. This is an example of complementary pairing of transfer RNA anticodon with the mRNA codon. So the messenger RNA, remember, that's the guy that actually went into the nucleus and read the DNA blueprint and translated it, and now it's got to transcribe it. So the transfer RNA anticodon is the opposite of the messenger RNA codon. Transfer RNA and messenger RNA still have a 3' and a 5' end. So the transfer RNA is going to read the messenger RNA codon 3 bases at a time from the 5' end to the 3' end and start building that chain of amino acids so our body can have the proteins that it needs. Woo woo. All right, there are 20 different amino acids, and there are 64 possibilities on how those amino acids can come together. There are start and stop codons. Typically, the start codons are ATG and GTG, and the stop codons are TAA, TAG, and TGA. So ATG is alanine, threonine, and glycine. So in prokaryotes, we don't have the DNA being housed inside the nucleus. It's just freestanding, free-floating out in the center of the bacterial cell. Well, I mean, it doesn't even have to be in the center, but, it's just a circular piece of DNA. So protein synthesis is constantly happening. If you think of it from a microbial perspective, the faster the prokaryote can make these proteins and amino acids, the faster it can grow or proliferate or make more copies of itself. So the whole purpose of this process is to get from the double-stranded DNA genetic code into the single-stranded RNA code, which will end up making a protein for that cell to use or for that body to use. All right, guys, that's all I've got. I hope that helps.